All right, everyone, welcome and thank you for attending the 2020 Macatawa Watershed Annual Meeting. My name is Kelly Goward. I'm the Environmental Program Manager at the Macatawa Area Coordinating Council. If you're not familiar with our organization, the MAC, we are a metropolitan planning organization for the Holland Zeeland urbanized area. We facilitate transportation planning for the region and then other issues of area-wide concern like the Macatawa Watershed Project and other issues as well. So tonight we are going to hear from 3 groups of Hope College students about some research they've been working on this semester. There will be time for questions at the end of each presentation, so please put your questions in the chat box and I will facilitate asking those questions at the end of each. After the student presentations are finished, we are going to have some networking time and split off into breakout rooms, and I'll provide more instructions about that later. After that, we will come back and hear some updates from Project Clarity, from the MAC, and from Grand Valley on the monitoring in the watershed. So at this time, I'm going to go ahead and turn it over to the students, and they will introduce themselves and take charge of the screen.
All right, hello everyone. My name is Kaylee DeRue. With me here I have Helen Holt, Jacob Vanderoost, and Christian Lundy. Oh. The topic of our presentation is surveying the presence of heavy metals in Lake Macatawa sediment samples. So starting off in August this year, we were trying to decide what we wanted to do for our research, and we came across a paper written by Dr. Peterson and several other research students in Hope College about heavy metal concentration studied in fish species in Lake Macatawa. Their study actually found high concentrations of selenium and chromium present in the fish. So our group was curious to see if similar results could be found within the sediment of Lake Macatawa. Lake Macatawa is about 175 square miles. It has a history of industry, which is a source for metals such as lead, cadmium, chromium, arsenic, and selenium. Our focus was on the eastern side of the North River Avenue Bridge, close to Window on the Waterfront in Holland. We focused on this area for a few reasons. One was because past studies have focused on the western portion of Lake Macatawa, and another reason was to compare the north and south shore in this area. So the north shore is used residentially, whereas the south shore is primarily has a history of being used industrially. As you can see, this is actually a Sanborn map from October of 1916. And this yellow area down here was actually used like for coal, was like a former coal yard. And this right here was actually a Hope Chemical Company. So our hypothesis was that there would be more of a heavy metal concentration in the soil on the southern portion of Lake Macatawa than the northern residential portion. So our project purpose was to measure the concentration of lead, cadmium, chromium, arsenic, and selenium, which are 5 of the Michigan 10 heavy metals in lake sediment samples along the north shore and south shore of Lake Macatawa as a continuation of Dr. Peterson's heavy metal research.
The main questions that we wanted to focus on during our research were which metals out of lead, cadmium, chromium, arsenic, and selenium are present in Lake Macatawa sediment and in what concentrations those are found. Our next question we wanted to focus on was, are the concentrations of heavy metals dependent on their location within Lake Macatawa, and are there patterns between the north and south shore? And our final question was, are the metal concentrations at a level that should be concerning for the health of residents of Holland? So this is actually a map of our sampling locations. As you can see, we did 3 sites on the North Shore, which is the residential area, and we also did 3 on the South Shore. which is the more industrial area. And we also did a terrestrial sample as a control to compare the concentrations to. And these are just some pictures from our sampling campaign. On the far left, that is Kaylee on the South Shore doing some of the sampling. In the center there is myself, a picture taken by Jacob on the North Shore of one of our locations that we sampled. And then all the way to the right, we have the top photo. Afterwards, we got a little wet, but it was well worth it. And then also on the bottom right is a picture of us organizing our samples. And then in the center at the bottom is actually a picture from, I believe it was North Site 2. And that's where we actually found some trash in the water at that location. So we just decided to note that. But these are all pictures from our field sampling location that we did in September. And so after we collected those samples, we stored them in a fridge just to keep them from growing any mold or anything like that. And then to prepare them for the microwave digestion and ICP, which will be talked about in a few minutes, we actually dried the samples in a simple oven here. So that's another photo of us drying our samples.
So before we could analyze the concentrations of heavy metals with inductively coupled plasma mass spectrometry, also known as ICP analysis, we had to go through a process called microwave digestion, which is a chemical process that dissolves the heavy metals into an aqueous solution. So none of us had any prior experience in microwave digestion, so we employed the help of Dr. Natalia Gonzalez-Pesh, who informed us that we should look for literature precedents on how to conduct this microwave digestion process. So we dug through some analytical literature, found this helpful literature precedent that gave us a procedure for microwave digestion. Building off of this literature, we conducted the procedure in which we put our dry sediment samples into these cartridges featured on the left-hand image. Then we added concentrated nitric acid, diluted them with water, and then cooked them at roughly 350 degrees Fahrenheit for about 15 minutes. After this microwave digestion procedure was finished, our samples were ready to be analyzed with ICP analysis.
So we took these solutions of heavy metals and nitric acid and we measured them using ICP spectrometry, and where they were measured against standard solutions containing our analytes. And we did this with the help of Dr. Jonathan Peterson from the Hope Geology and Environmental Science Department. And our results. So for all of our sites, we found the levels of arsenic were below the detection limit. So we couldn't actually tell what the amount was. For lead, we got higher values, but for most of these, they were below the limit of quantification. So we could tell that there was some of this analyte there, but we couldn't be confident in the amount. For the last site, Site 3, we got a high enough value we could quantify it. So that was some measurement there. For cadmium, we had pretty low levels as well, but again, with one site that had levels above the limit of quantification. For chromium, we had higher values all around. Every site had a value above the limit of quantification. And for selenium, we had mixed results. Some sites had values below the detection limit, and others had values above the limit of quantification, and those ones were significantly higher.
This is a plot indicating the levels of chromium at the different sites. So T right here is indicative of terrestrial, N represents the North Shore, and S represents the South Shore. So here we can directly compare the concentrations of the analytes depending on the location. So chromium was quite notable in the sense that you can see here based on these graphs that the North Shore featured higher concentrations of chromium comparatively to the more industrial South Shore. Looking at cadmium, there was a large cadmium spike, specifically in the first location of the South Shore. Any of these locations without any bars indicate that the detection of the analyte was below the detection level. So cadmium seemingly had higher concentrations on the South Shore. Selenium similarly featured higher concentrations on the South Shore comparatively to the North Shore. The North Shore only had 2 sites that were above the limit of detection. And lastly, lead also had notably higher concentrations along the South Shore comparatively to the North Shore. So it seems our hypothesis seems to kind of fit the bill when looking from the North and the South Shores.
So doing some further analysis into our results, we compared our observed values to the standards set by the state of Michigan and their groundwater surface water interface protection criteria, as well as the Environmental Protection Agency's screening levels. And both of these standards are set to ensure that the heavy metal levels don't exceed what would be a health concern and pose an environmental hazard. So for all of our analytes, except for selenium, the levels were far below the Michigan standard and the EPA standard. However, for selenium, the highest observed value was 0.745 parts per million, and the Michigan standard for the protection criteria is 0.4 parts per million. So that was once a point of concern for us. So diving deeper into this selenium concentration, the purple value shows the Michigan screening level, and which is 0.4 ppm. And 4 of the sites have a value above that level, with 3 of those being on the South Shore. And so some conclusions we drew from this: the observed levels of arsenic, lead, cadmium, chromium are below the respective levels considered to be environmental health hazard for humans. And the measured selenium concentrations on the South Shore were greater than the Michigan standard, but less than the EPA standard. And except for chromium, the metal concentrations on the South Shore were greater than those along the North Shore. And we hypothesized this difference in concentration could be attributed to the historical activity in close proximity to the South Shore of Lake Macatawa.
Potential future work that we could do to build off of this project would be comparing our ICP results with atomic absorbance spectrometry to kind of cross-track our analytical results. We can perform the same analysis on sediment samples from the middle of Lake Macatawa, or on a deeper section of the sediment cores to try to see the gradient as you go with depth to see if there are any patterns with heavy metal concentrations. We can conduct further literature investigation to determine specifically why the South Shore features higher metal concentrations. Our initial hypothesis assumes that the industrial activity of the South causes this, but we could always look further. We can investigate potential sources of chromium along the North Shore. If you remember, chromium actually had higher concentrations along the North Shore than the South Shore. So we tried to investigate the specific reasons why that was found. We could determine connection between the high concentration of the selenium in the lake sediments with the high concentrations of selenium in the fish from Dr. Peterson's study to see if there's any correlations or connections between those two results. And lastly, we could conduct a similar experiment comparing the east and west rather than north and south to see if there's a gradient of heavy metal concentrations going from the east and west. Here are our references for this project, and we are happy to take any questions at this time. Thank you very much.
All right, I assume everyone was virtually applauding from home. So I do have a couple questions in the chat box, and feel free to continue adding them if you have them. If we don't have time for all the questions, we can get written responses from the students and provide those later. So the first question is, how does the Michigan selenium standard compare to the limit of quantification your method achieved?
I'll go back to the chart. Yeah. So specifically comparing our highest observed selenium value to the Michigan standard and how those compare. Is that the question?
No, the detection limit.
Right.
Oh, that's compared to the detection limit.
I'll answer this.
Yeah, go ahead. That's tough side.
I believe the limit of quantification is around 0.2 parts per million, which is a little bit lower than the Michigan standard.
Okay, perfect. Second question, how were the sediment samples collected, and how did your collection procedure take current and historical pollution into account?
Yeah, so our collection procedure, originally we were going to use a dredge, but we quickly found out that that was not going to work for us just based on the one that we had. So we actually used a shovel and pretty much just got surface sediment from Lake Macatawa. So that was our main sampling procedure to get the sediments. And what was the second part of that question as well?
Yep, so the second part was, how did you take into account current versus historical pollution locations?
So we really just focused on the historical pollution, because as we mentioned before, the site now is close to a window on the waterfront, which is actually a park area. So as you can see, most of this on the south shore here is green. So that's actually not industrial at the moment. So we really just focused on the historical industry in the area.
And then,
Okay.
the depth that we collected with the shovels, the results we have are more indicative of the more contemporary sediment samples that have been laid probably in the past decade or so. So if we wanted to really get down to the sediment samples that were deposited during those more historical periods, we'd have to sample a little bit deeper with sediment cores of some kind.
All right, and one more two-part question. Do you have suggestions from the literature as to how patchy the metals might be expected to be? Are shoreline metals site-specific, or are they likely to be scattered by lake or stream processes?
Yeah, I would assume that they would be scattered just by the way that the Macatawa River interacts with Lake Macatawa. So again, as you can see in this photo, this river area, kind of swamp area, is ever changing. So a lot of it is spread out and moves quite frequently.
Yeah, especially the fluvial flow of the Macatawa River would probably be more conducive to a more spread out distribution.
Okay, excellent. I don't have any more questions. Thank you to the students and thank you to those asking questions. I did prepare the students that this group usually asks some tough questions, so you did not disappoint so far. So with that, we'll go ahead and pass it on to the next group of students.
Thank you.
Thank you.
All right, so my name is Madison.
I'm Dominic.
And then I'm Erin.
All right, so our presentation is on the determination of heavy metal accumulation in earthworms, specifically Lumbrius rubella collected from the Macatawa Watershed. So the purpose of our experiment was to analyze the heavy metal concentration of cadmium, lead, nickel, and chromium in earthworms within the Macatawa watershed to estimate the potential exposure risk to the environment and the ecosystem via biomagnification. So first, why did we look at earthworms? Earthworms, first off, play an integral role in soil formation. They are an indication of healthy soil. So as they interact with the soil, they make holes for aeration or for water. And they're also typically prey for terrestrial organisms. So we would assume with biomagnification that the metals accumulate as they go up, which I'll talk about in the next slide. So to discuss bioaccumulation versus biomagnification, when we are looking at the worms, we are focused on bioaccumulation because that's all we can detect from the worm specifically. We use the data we collected from the worms to make some assumptions about how the metals could potentially biomagnify. And when we chose the metals we were going to analyze within the earthworms, we chose metals that, based on the literature, biomagnify. So biomagnification is a process in which a compound increases its concentration in the tissues of an organism as it travels up the food chain. This means that as the earthworms interact with the soil, they gain the heavy metals in their tissue. And when a predator of an earthworm eats the earthworm, the heavy metals within the earthworm are accumulated by that predator. And as you go up the food chain, it gets more and more.
So we obviously we chose these metals because they bioaccumulate and they biomagnify. However, we also wanted to take a look at what kind of damages they can cause to the ecosystem and human health as well. So just to start off with cadmium, it can cause damage to respiratory, renal, skeletal, and cardiovascular systems. And it can cause several varieties of cancer. And it can be ingested by eating food contaminated with cadmium and also by smoking cigarettes, which it's located in the filters of cigarettes as well. So when people toss their cigarette butts out the windows of their cars or wherever, it can leach into the soil and then be introduced into the ecosystem that way as well. Lead as well damages the kidneys, brain, skeleton, and nervous system. You can ingest lead through inhalation of contaminated air or contaminated food or water, just like cadmium. Nickel is very similar to lead in its exposure pathway, but it can also cause dermatitis. A lot of people are allergic to it. It can cause organ diseases and cancer of the respiratory system. Chromium is also highly toxic and a carcinogen. It's associated with slow healing ulcers, and it can destroy DNA within your cells. You can also be exposed to it through inhalation of contaminated air, and it's also a water-soluble compound, so it can be in water as well. So the questions we sought to answer with our research was, what are the concentrations of the heavy metals, cadmium, lead, chromium, and nickel in our earthworm samples? How do the sublocations that we chose for our sampling sites correlate with one another? How do the sublocations correlate with both human exposure and ecological impact? And which one potentially has the most risk associated with it? And then we also wanted to take a look at the biomagnification risks and the ecological and human health implications that are associated with our heavy metal concentrations. So we sampled 11 different locations, and we wanted to get a really good look at just 4 different sublocations that we thought would be provide some interesting data. So we tried to look at sites that were previously industrial in nature. We tried to look at some more natural areas as well, without a whole lot of human interference and contact. We looked at residential areas, and then we also wanted to look at agricultural areas to see what kind of heavy metals are present in the soils where we may be growing our food or growing the food that our food eats. So to start off with our industrial areas, we explored Collin Park, which is about a half mile wide, and we chose the north, east, and west sides because they're all 3 of them are located directly in between 2 active industrial plants. For agricultural areas, the people over at Alot Farms were kind enough to give us free reign to sample any of their fields. So we chose a soybean field and 2 corn fields, and we went ahead and dug up worms, and there's Madison there digging up the worms. We also chose a couple of natural areas, mainly Stuyvesant Trails. It's a wetland area with a lot of possible leaching potential there, as well as Quincy Park. When I went there were a lot of kids running around playing soccer, but Quincy Park is located right next to a former dump site from a few years back. So we wanted to take a look at some of the heavy metal concentrations there. And then with residential areas, we just picked some simple ones like the skate park right on the east side of Holland. And we also picked Aaron's house and Madison's house because they're both right in the middle of large neighborhoods in Holland. And for our sampling methods, we went out usually after a strong rain a couple times a week, and we would collect about 10 to 15 worms at each sampling location. Once we had collected about 10 to 15 worms, we called that good enough. We brought them back and we labeled them. We placed them in crucibles. And then we wrapped them in foil for fridge storage. So we stored them in the refrigerator for about a week to give them time to excrete their soil contents, because remember, we want to test the heavy metal concentrations in their tissues and not what's in their guts.
So for our metal analysis, we chose to do atomic absorption spectroscopy, and we began by digesting the worms through various chemical processes. And then we filtered the solution to remove any large particles so that it could go through the AA machine. And then we prepared our stock solutions of cadmium, lead, nickel, and chromium in concentrations of 0.5, 1, 5, and 10 ppm. And we chose those concentrations based on literature values. And then we performed our analysis. And then in the picture on the left, you can see where the worms are being filtered, and then where Dom is using the A machine to get our values. And then here's just a picture of our worm solutions after they've been filtered. You can see they're like a clear yellow color, and then our standard solutions on the right. And then here's just a table showing the different criteria, safe levels for the metals that we chose. We chose, like, the WHO was one of the first ones that's in there, and then we brought in some Michigan criteria and then New Jersey, and you can see they're different between each states, but all of our values were found to be within safe limits. So this is cadmium. We can see elevated cadmium levels in residential sites 11 and 2. The BDL is below detection limit, so unfortunately we were not able to detect below 0.02 ppm. And then this is the lead concentration. Again, it is within safe levels, but the elevated sites are 10, 11, and 2 and 3. So that's again our residential and industrial sites. And then nickel concentration, we didn't have any BDLs here, but we did find some elevated levels in the residential areas at 9, 10, and 11. And then again, with industrial site 3. Chromium didn't have much data to go on here, but there are no sites of concern, and it is again within the safe detection limits. So soil concentration versus bioaccumulation, what we found is that metal availability for bioaccumulation is heavily influenced by soil characteristics. So the pH of the soil, the oxidation state of the metal, the solubility of the metal, etc. So there's no real way to like apply other literature values to our, like say the ratio of metals that have been accumulated in worms to the concentration in the soil, because they're all so different. But according to the literature, there is typically a strong positive correlation between the proportion of heavy metals accumulated in worm tissue to concentrations of heavy metals in contaminated soil. So we infer that the concentration of metals in soils is typically higher. So we would expect the worm concentrations to be decreased compared to the soil. So our conclusions, so how do the sublocations of sampling sites correlate with one another? We found that residential and industrial sites were determined to have elevated levels of nickel, lead, and cadmium. However, nothing was outside of the safe detection limit. And then how do the sublocations correlate with both human exposure and ecological impact, potentially, which has the most risk associated with it? So again, the most risk associated would be with industrial and residential sites, as they were the highest. And then what are the biomagnification risks and possible ecological and human health implications associated with these heavy metal concentrations in worm samples? And we said there was little risk associated, as all concentrations were within safe detection limits.
So for seeing how we would want to take this project in the future, if we had further ways to go with it, first we'd like to repeat our experiment. Try different methods of analysis, such as ICP, as the previous group did. We'd also want to obtain a larger sample size and test both the worms and the soil, so that we could compare the two and see how much less or more are in the worms in comparison to the soils for heavy metals. We'd also like to test other metals that we saw in the literature, such as thallium and methylmercury and arsenic. And we also wanted to, but due to time constraints, we were hoping to test for DDT. So that is something we'd like to do in the future as well. These are our references. We'd like to acknowledge the Hope College Geology and Environmental Science Department, Professor DeVries Zimmerman for advising us throughout our project, Dr. Peterson, who helped us use the AA machine for our analysis, Dr. Brown advising us how to digest the worm samples for analysis in the AA instrument, and Al Farms, which allowed us to sample on their property, which were our agricultural sites.
All right. Awesome. Again, everybody's giving you a virtual round of applause. Have one question that's come in so far. Actually, I guess a two-part question. So any other questions, go ahead and type them in the chat box. So the first question is, were all the worms gathered confirmed to be the same species? And did you do any literature research to see if worm species varied geographically?
I can do that one. So we took a look at, there were only 3 main species that occur kind of in the Southwest Michigan area. We were finding largely the same species. Very few were of the larger variety. Most of them were small, kind of what you'd call like your typical red worm. And we were taking them, and in the end, we were doing this based on mass. But nothing in the literature suggested that they would uptake different amounts. It wouldn't vary between species. We think it would just have to do with overall exposure.
Okay, another question about worms. Do you think that the age of the worms impact your results? Did you grab worms of similar size to account for that?
Again, I would say, yeah, if there were older and larger worms, they probably would have had time to accumulate more metals over time. Largely, we grabbed roughly the same size worms. When we were struggling to find a lot of worms at a site, we would take some smaller ones as well. But in the end, we wanted to make sure we had enough that we could weigh them on a scale and get a good mass, and then be able to work from there mathematically. So to answer your question, yes, I would say the age of the worm may matter. We tried to take that into account, but also sometimes we just needed to make sure that we hit the proper amount that we could make sure we can measure them and produce some results.
All right, and did you examine soil pH or notice any striking differences among the soil samples that might indicate differences in bioavailability of the metals?
No, we didn't have, due to time constraints, we weren't able to examine the soil around the sampling sites, but that would be like in the future, if we were to continue this research project, that would probably be where we would go with it.
All right, excellent. Thank you again, students and audience members, for your questions. So now we'll move on to our third and final group of students.
Thank you.
Thank you. Hello, so my name is Kate Kim. We have Alex Vasquez and we have Kaylin Lagerwald joining us online here. So our project was focused on the assessment of macroplastic pollution and storm drain discharge due to rain events, specifically in the Holland area. So first we had to answer the question of why plastic pollution is important. And so just looking from the time between now since the 1960s, we've had such a dramatic increase of plastic production, but a very small recovery rate of that plastic. And a lot of it ends up in marine debris, as you hear, like 80% of land-based sources, or land-based sources account for 80% of marine debris. And unfortunately, unlike what we would hope, plastic does not disintegrate or break down as we would like it to. So break down until you get to a microscopic level to the point that it's very hard to retrieve, and it just stays in the environment and it causes issues. So we chose this project because there was some studies done at Hope College before, but they were more focused on microplastics, so that small type of plastic that's hard to recover. We decided to look at the macroplastics, so the bigger pieces of plastic that you can pick up and collect. And that would give us an understanding of the pollutant and then understanding potential sources. So what we wanted to address was, is plastic pollution present in storm drain discharge in the Holland area? What is the most common type of plastic pollution? And then what's the size and state of the plastic in the discharge? Then how do major rain events affect the amount that we're going to collect? So we had two sites in particular, and both of them were storm drains. So the two arrows are the two different sites, and those are two different storm drains that we collected from. So they're very similar in some ways in terms of the environment that they're in, but they do have some notable differences. So we have Brindle on the waterfront. So the arrow there is pointing to the storm drain, and it's more of like a secluded area. And this storm drain runs from Columbia Avenue and comes out here. So we don't necessarily know where the pollution could potentially be coming from because it's not clear. But this is where we would collect, and as you can see, it's a pretty steep drop off in terms of the ledge there to collect. And then the 22.3 meters, just the length of the area that we decided to collect from. The second location was Visitor Farms, and that is me. So the blue arrow is pointing down into the storm drain as well. This actually is located right on Chicago Drive. So this has a very easy access from a lot of traffic, a lot of people. So yeah, so we have two different sources of pollution potentially. So in order to kind of try to figure out our research questions, first we wanted to go out once a week using the vegetation as a natural barrier. So we would go out with the big plastic garbage bag, and we would collect as much plastic as we could from the designated amount of area that we decided. And so we collected everything that we could, and then we were separated. Once we got back into the lab, we were separated into different categories based off the type of plastic it was. So you have your beverage type, which would be the cups, water bottles, soda bottles, your packaging, your candy wrappers, chip bags, whatnot. Food and drink accessory, you have your straws, cutlery, bags, you have your grocery bags, garbage bags, etc. Then other, that can be anything from like a plastic flossing tool, etc. So this is an example of something we did.
And so each piece of plastic was weighed in grams using a mass balance scale. And then measured of its longest length in centimeters and using a ruler. The number of rain events and the amount of rainfall that occurred the week prior to sampling was also recorded using data from the Tulip City Airport. So this table shows the total number of pieces of macroplastic collected from each of the sites during each of the sampling that was conducted, which turned out to be once a week for 5 weeks. And so Visitor Farm experienced overall we found more plastics at this Visitor Farm site totaling at 237 pieces versus the Window on the Waterfront site, which totaled at 165 pieces. And here's the number of pieces versus date for both of the sites. And so the most amount of plastic was found on September 24th. And this served as our background for the analysis, for we're not sure how long plastics had been collecting at the sites prior to this date, but from then on, plastic was collected with only a week in between, and we still observed a decrease in the amount of plastic found over time. And these images show this phenomenon of how plastic decreased over time versus October 1st in the left-hand pictures and October 14th on the right-hand from the Window on the Waterfront site, which just goes to show how the number of pieces had decreased. And we also thought it was important to note broken down grocery bags and microplastics that we found when conducting sampling. And these pieces were not recorded in our results because they measured less than 5 millimeters in size, which at that point they're considered to be a microplastic, but it's still important to note their abundance in the watershed, and we observed them even when doing our sampling. And here you can see the overall type of plastic for both of the sites. Packaging was the most found, totaling at around 150 pieces, followed by bag, other, food and drink, and beverage. And this information could prove to be very helpful in awareness campaigns and plastic pollution mitigation, just to understand what the most common type of plastic is, or at least we were finding in the environment. And here is the overall average weight versus date for both of the sites. And so the average weight of plastic ranged around from 4.3 to 2.6 grams and remained fairly consistent over time. And here's the overall average length of the plastic versus date for each of the sites, which was about from 15 to 10.2 centimeters in length. And given the average weight and the average length that both of them stayed fairly consistent over time, we were able to note that there's an average, like, continuous flux of plastic coming in. But yeah, it was the same over time, at least from in these terms of weight and length.
All right. So the last kind of research question we looked at was to see how major rain events would also affect the macroplastics. And here is a graph looking at the overall amount of pieces. So we don't really see any sort of significance in line with the amount of rain. Here is the total weight, though, for each of the sites and corresponding with the amount of rain. And it's important to note that even though we don't really see significance, the weight kind of stays similar. So it's like, even though, you know, we might be getting less like number of pieces, we're still getting kind of a constant weight coming in each week. So it's also important that we look at the climatology report. This is for the month of September. We see that the precipitation total for this year in September was about 3.06 inches, when typically it's about 3.9. And this is lower than typical. So unfortunately, looking at the rain was less than kind of what we were hoping for the rain event aspect of our report. Here is for the month of October, and it had a little bit more than typical, but I mean, even looking in comparison to last year, there was 6.31 inches. So unfortunately, the whole major rain event was tough for us to see here. So like I had mentioned before, the average weights are kind of similar. And even though 3.41 grams for the two outfalls weekly seems kind of small, if you look at this on a 10-year scale, that's 4 pounds, and we are only looking at 2 sites. So, you know, however many sites there are total in the Macatawa watershed, it's important to note that there is a lot more than we think. And here's a picture kind of showing what 3.41 grams look like, even though it doesn't seem like a whole lot when looking at the weight. It does, you know, accumulate quite a bit with the larger pieces. So kind of going over our conclusions. So it's definitely, pollution is definitely a problem here in the Macatawa watershed. Looking at both sites that we did find that the packaging was the most abundant type of plastic pollution. The average weight ranged from 2.6 to 4.3 grams, and then the average length was 10.2 centimeters to 14.46 centimeters. So like I was saying earlier as well, with the rainfall, even though we didn't see any significant correlation, it's definitely something that should still be considered with months with more major rain events
And then it also is important to note that we are still getting constant fluxes of pollution from these macroplastics each week. So kind of looking at like what's next. So it's clear that there's an issue. We did only look at the 2 sites. So looking at more sites around the watershed is very important. I did read an article that Washington, D.C. in particular has a taxation on their plastic bags now. And before this, they were kind of averaging about 22.5 million bags a month. And after they implemented it, they're only seeing about 3.3 million bags. So that's, you know, even though it's a, still looks like a large number, that anything helps at this point. And then another thing is stopping the purchase of bottled waters. You know, reusable water stations in certain areas around the city can be very helpful. And, you know, in times of COVID, it can be kind of hard to look at that as a shared station, but when we kind of defeat this virus, that is definitely something we can also look at. So those are our references, and if anybody has any questions, we'd love to answer them.
All right, thank you. So I do have one question that came in. Is it assumed that the plastic recovered was delivered by the storm drains?
So initially we kind of looked around the areas on the window on the waterfront. There was that, it was kind of on the slope there. So, you know, whether that came in from like outside sources. So it wasn't assumed. Originally we were going to assume that it came in, but just kind of the circumstances that we had, we just kind of did around. So no, not assumed.
All right, and the next question, I think related to this, did you sample plastic floating on the surface in proximity to the storm drain, or was it trapped in some structure at the mouth of the drain? Also, how thorough do you feel you were at clearing the area during each collection visit?
So we typically were collecting plastics that were found around the storm drain, so like caught in the vegetation or stuck in the dirt. And in terms of our whether or not we collected everything, I say we did a pretty good job considering there were 3 people going around. We try to make sure that it was a manageable area. And I mean, there were times we would double check, triple check, to make sure we got everything. So yeah, I would say there would be a good chance we've gotten at least a good majority.
And I'd also like to add in, we used vegetation to collect, like stop the plastics and help them to collect, but it is also possible that the vegetation did hide some plastic from our site.
Okay, excellent. That's all the questions we have. So thank you very much. Thank you again to all of the students. I think you all did an excellent job.